all, and good afternoon. Hope you are going fine and staying safe also. So I welcome you all to this talk on the topic research challenges in bioinformatics. So here's a brief outline of my talk. So I'll be mainly talking about the contemporary and emerging trends in bioinformatics. In any case, I'm not planning to give an exhaustive list of challenges. I just plan to give a flavor of the major areas in bioinformatics where a person who is interested in doing research can concentrate. So I'll begin by quoting Professor Donald Knuth, the celebrated computer scientist and professor at Stanford. So the number of radically new results in pure computer science is likely to decrease, while scientists will continue to work on biological challenges for the next 500 years. So this was his words in 1993. So surprisingly, this has come true. So indeed, scientists they have always been fascinated by the amazing complexity of the living organisms. And right from the invention of microscope to the latest advancements in bioinformatics, such as the stem cell research or the innovation of artificial intelligence techniques in computing, or the personalized medicine concept that we often see in press today. So all these have been different attempts to unravel the mysteries of life, unfold the excitements in life science. So in fact, bioinformatics as a science, you can define it as an interdisciplinary area or field which involves the study of properties of living beings from various perspectives, that of a biologist, that of a computer scientist, and that of a statistician or mathematician. In fact, the research in bioinformatics can range from very simple mathematical modeling of the biological properties of a system to development of complicated algorithms and data analytics tools for analyzing biological data. So a fresh graduate in bioinformatics can expect to work in any of the areas like teaching, research, consultancy, or even clinical service. So in fact, the scope of bioinformatics is not restricted to any of these areas. And with the advent of high throughput bioinformatics tools and the advancements in data analytics, big data and data analytics very recently, so there has been an unprecedented increase in the amount of data that is around various databases across the globe. So this uprise in the huge amount of data has enabled us to analyze and study the properties of various organisms from a data mining perspective or data mining point of view. But at the same time, it has thrown in quite a few challenges also. So I would like to classify these challenges into contemporary and emerging or futuristic. So I would quickly walk through the contemporary challenges first, and then I will move on to more advanced emerging challenges or futuristic thoughts. So I just want to know whether I am audible and the slides are visible.
Yes, sir.
Okay, thank you. So pattern matching has been an area of interest from the very beginning. So pattern matching involves the mining of significant patterns in the DNA. So at a very simplistic view, if I give, the DNA is actually like a bead which is strung in a particular thread. So the beads that constitute the DNA are 4 nucleotide bases, which we label as A, C, T, and G. They stand for adenine, thymine, cytosine, and guanine. So these nucleotide bases form the basis, or they form the building blocks of our life. So disease networks or disease progressions are often associated with aberrant patterns of this ACTG sequence. So if we could mine using some data mining algorithm, if we could mine or identify some co-occurring and frequent patterns in these networks, then it may help in disease diagnosis and it may help in identifying the progression of a disease. So with this view, people have been working in this fundamental area, that is pattern matching, for years, and now it has almost reached at its zenith. So it's now people are concentrating on cancer bioinformatics. So it has emerged as a separate branch in itself. So it basically deals with detecting cancer biomarkers from DNA sequences and by analyzing the expression patterns of genes. So we'll come to that later. Now another one is the analysis of next generation sequencing data. So with the advent of high throughput sequencers and bioinformatics tools, now it is possible to sequence. Sequencing means decoding the genome. So there are about 3 billion nucleotides per chromosome, or per DNA. So if you could decode all these things, it would be a very big data base. So it's not possible to write it or enumerate it in a piece of paper or in a particular file. So the surprise is that even though all the cells contain exactly the same set of genes, they behave differently. A neuron behaves quite different from that of a muscle cell or from that of a blood cell. So why is it that so? Even though all the cells contain the same set of genome, the behavior is completely different. Why is that so? This is because of the changes in gene expression. So they may be expressing it differently. The set of genes that are expressed in a neuron may not get expressed in another cell type, say blood cell. So when we say express, we mean that the gene is actively producing a protein. That's it. So when a gene actively produces a protein, we say the gene has expressed. Otherwise, the gene is dormant, or we say it is. We can say it is hidden, or it is not expressed. It is dormant. So if we can quantitatively measure the gene expression levels or behavior of the genes, then we can identify the difference in behavior of cells, or difference in behavior of pathways, or how they differ across a normal and a patient with a cancer, or patient with some other disease or abnormality. So this forms the basis of techniques or research methods which analyze the next generation sequencing data. Then you have the pathway analysis. So by pathway, we mean a network of interconnections of genes. So genes do not act individually. They often interact with each other to form a network of interactions. That network, we call it as a pathway. Just like a graph in computer science, this also is a graph. The vertices are genes, and the edges are interactions between the genes. So systematically, if we analyze the pathway, we can get to know about new diseases, or what type of unwirings or what type of rewirings are happening in a particular network. So these rewirings can help us to learn about the progression of disease. We could detect a particular stage very early. We could detect a cancer at a very early stage. So these kind of analysis can be done if we analyze the pathways very carefully. We follow the pathways very carefully. So that is pathway analysis. Then you have sequence alignment. So all these are traditional methods, contemporary methods. The research is already happening, or people are already working on these challenges. There are lots of papers by which you can get information about. Then sequence alignment. So as I mentioned before, the DNA is actually a sequence of nucleotides. So if we could enumerate the entire genome, then it could run some 6 or 7 times the distance of the moon and Earth. So computational genomics deals with a large amount of sequence data. So most of whose functionality and structure are unknown to the scientific world even today. So if we want to say, for example, characterize the ethnicity, or say, for example, characterize the ancestry of a particular individual, then there is no other way than finding the functionality and structure of the sequence. Maybe his sequence may be having some similarity with an ancestral sequence. So for that type of phylogeny analysis, we need to understand the functionality and structure of the sequence properly. But unfortunately, most of the sequences that we find out today, their functionality is not known exactly. So what all we can do is to ascertain the extent of similarity with a known sequence. Maybe there are some known sequences which are close enough to the human sequence. So we can ascertain the extent of similarity with a known sequence. That known sequences are called reference sequences. So there are public databases which provide curated, highly curated and reference and publicly available reference sequences. So we can analyze these publicly available reference sequences for the functionality of the unknown sequences. So we can have some global and local matchings, and by analyzing the matchings, we can get to know about the what the percentage of similarity between the unknown sequence and the known sequence. So there are several publicly available tools also. To mention a few, ClustalW, then BLAST, basic local element, BLAST. So all these are publicly available tools which you can use for performing sequence alignment. So enough literature is already there in this area. Then you have the protein structure prediction. So it's a very classical task. So the nucleotides, they actually, or the DNA nucleotides, they actually are translated into a structure called proteins. So they are not directly translated. They are first transformed in an intermediate stage. That intermediate stage consists of 3-letter codons. The structure is called codons, C-O-D-O-N, codon. So these 3-letter codons are then mapped to proteins. So while this mapping happens, it is not simply that a 3-letter codon is directly mapped to a protein. So they form different type of structures. They form different type of foldings or 3-dimensional structures, which are impossible to view in a 2-dimensional plane or using a 2-dimensional tool, a traditional computer graphics tool. So in order to predict the 3D structure of proteins, given an amino acid sequence, so they are composed of amino acids, so which are molecules formed from the bases. It's actually a chain of molecules. So in order to predict the function, we need to first understand the structure of the protein, because structure and function are closely related. So in order to predict the function, we need to first explore and visualize the structure of the protein. So for that, there are several visualization techniques. So complex structures are mostly represented by using graphs, data structures like graphs, cubes, trees, maps, and all. So these kind of visually appealing structures are nowadays available. So different data exploratory tools are available in statistical packages like R, Python, and all. So even some of them like RASMOL and PyMOL, they provide interactive data visualization. So we can interact with the data. That means they will provide a handle. We can use that handle to rotate the object and view the other side. So we have seen such kind of tools in maybe in other areas. So these are some contemporary ideas. That is the challenges which are already in place. Active research is happening. So as I mentioned before, this is in no way an exhaustive list. We can find out many other areas, related areas like natural language processing, image processing, which share frontiers with the bioinformatics. But still, I have consolidated some of them. That's all. Now I move on to some futuristic ideas, or these are emerging areas which promise active research in the emerging years to come. So first one is epigenetics. So epigenetics involves the study of inheritance mechanisms that do not involve changes in the DNA sequence directly. So you can have an example like bee. So there are different types of honey bees, right? Queen bee and a worker bee. So if you take a beehive, you have queen bees and worker bees. So the genetic makeup of the queen bee and worker bees are exactly the same. Their genetic makeup, their structure, everything is exactly the same. But of course, there are slight differences in the structure. If you could see that they are slightly yellowish in color, their abdomen is slightly elongated. So apart from that, there are certain things, certain chemical things or chemical modifications are there in their genome that makes a queen bee superior to the worker bees. So that such kind of questions, very interesting research questions. Or how, another question, how does a single-celled zygote develops into very complicated and sophisticated structures and organ systems in our body? So a blood cell, a nerve cell, then a muscle cell, all these are coming from the same zygote or same single-celled body structure. So how is this differentiation happening? So such kind of questions are often answered by mechanisms called epigenetic mechanisms. So this can be thought of as chemical relays, just like it's they are biological counterparts of our relay switches. So they simply turn some genes on or off. So turning a gene on means favoring its expression. We are favoring its expression, and that means we are providing enough opportunities for that gene to get translated into a protein. And turning them off means we are against its expression. We are we are disturbing its normal expression pattern. So such kind of chemical modifications often interfere with the gene regulatory mechanisms. So even though the exact mechanism of gene regulation is not clearly understood, but people are actively researching on epigenetics, and especially one commonly studied epigenetic mechanism is called DNA methylation. It is the addition of methyl groups or CH3 molecules coming and attaching to the DNA. So these kind of changes are being studied, followed by scientists, and we'll soon hope that the epigenetic mechanisms of inherit. And the interesting thing is that these are also inherited. These epigenetic changes or mechanisms are inheritable. They are inherited from the parent to the offspring. So such kind of mechanisms are soon elucidated. Let us anticipate that they are soon elucidated, and even aging, the emotional changes that are happening to elder people or due to aging and other disease conditions, they are also being closely studied in this purview. Then comes the non-coding RNAs. So previously there was a misconception that about the 95% of the DNA were junk and that they don't have any functionality. So the only portions that contribute to coding or protein formation are the rest 4 to 5%. But now recent researchers have highlighted that these junk, the so-called junk portions, also had some important biological functionality. So the RNA molecules which do not contribute in protein translation, which are called non-coding RNAs, they also had a very critical impact on gene regulation. So day by day, more functional non-coding RNAs are being discovered, and their characterization is performed. But still, we have to go a lot in this area. So functional analysis of non-coding RNA has been a very recent attraction. So over a decade, it has been just under a decade, it has been the point of interest. So people are working on functional analysis and basically characterization of computational modeling and characterization of the non-coding RNAs. People are studying the properties of non-coding RNAs in different perspectives. That is, some are long non-coding RNAs, then you have micro non-coding RNAs. So all these things people are studying nowadays. And it is also an epigenetic mechanism, because non-coding RNAs are also taking part in gene regulation. So in that aspect, can also be thought of as an epigenetic mechanism. Then I'm interested in molecular pathological epidemiology. So it is a very recent area, very emerging topic. Only after 2012 or 14, it has gained popularity. So it amalgamates research efforts towards integrating precise classification, molecular classification of cancer and epidemiology. Epidemiology refers to population. So whenever we hear the term epidemiology, we are thinking of population, a particular group of people. So in fact, it refers to the specific environmental conditions of a group of people. For example, you can think of the Nipah. Nipah has recently created a panic in certain outskirts of Kozhikode. So Nipah is not an epidemic or a pandemic, but it is attributed to the cause of that Nipah disease is attributed to an etiology. Etiology means the sum total of environment and habits of a group of people. So Nipah has been attributed to the eating habits of a particular bat. So whatever it may be. So etiology refers to the sociological conditions around a group of people. So this molecular pathological epidemiology studies or closely monitors the molecular changes in diseases. Especially they are concentrating on cancers now, because some cancers are thought of as etiology or related to etiology. So they are pursuing such changes with respect to a particular area or locality or etiology. So the impact of epigenetic changes happening at a molecular level are closely studied, and all these research efforts are consolidated under an umbrella called molecular pathological epidemiology. So I have included one or two high quality research papers in the reference section of this slide. So you can find that these are latest trends in bioinformatics. Then comes the interesting part, that is the deep learning and artificial intelligence in genomics. So with the innovation of artificial intelligence and deep learning, I think genomics and bioinformatics in particular has gone a great leap. So in fact, there has never been so much excitement of AI in bioinformatics. So particularly in gene prediction, deep learning is now applied. So gene prediction is an area that seeks to identify the regions of DNA that code for proteins. So as I mentioned before, not all regions in the DNA sequence are coding. So only some regions are active, and those regions which are called genes, and in genes itself, only some region of the gene is carrying the active what you call coding portion. The other regions are helping regions or some other regions whose functionality is yet to be deciphered. So identifying which portions of the genome are coding, or identifying which portions of the genome are the so-called coding genes, that's a non-trivial task. So people have done high-quality research recently, and they have come up with algorithms like DanQ, DeepGS, and DeepCPG. So these are actually, this DanQ and DeepGS are convolution-based neural network models that can predict the genome. They can predict the genes in a particular genome. So they help in identifying the regions that are coding. Similarly, DeepCPG is a convolution, and it's a hybrid of convolutional and recurrent neural network model, which helps to identify the relationship between DNA methylation, which is an epigenetic mechanism, and gene expression. So how this methylation affects gene expression, that is being studied, and they have named it as DeepCPG. Maybe it is because they are analyzing CPG, which is a region called island inside the genome. So CPG stands for cytosine phosphate guanine. So it's a particular bond, cytosine bonded with guanine with a phosphate bond. So that portions where the methylation phenomena is often seen are closely monitored for changes in gene expression patterns. So these are deep learning initiatives. And in my opinion, as of my reading and understanding, the best research that is happening, intensive research that is happening in bioinformatics and this deep learning are located in universities in Switzerland and Germany. So you can find interesting tutorials prepared by German scientists on how to analyze TCGA data using artificial intelligence techniques. So this TCGA is a project that is hosted by the Broad Institute, which stands for the Cancer Genome Atlas. The Cancer Genome Atlas. So it is a project that aims at revealing the blueprint of a variety of cancers. So I think some 40 different types of cancer pathways are being identified in TCGA project. So they are providing data for gene expression, DNA methylation, then pathway specific and some other kind of variation, genome variation data freely and publicly available for use in research. So also the Max Planck Institute of Germany and the Swiss Swiss Institute of Bioinformatics, that is SID. All these are class institutes which are pursuing high quality research in translational medicine and genomics. And recently, Microsoft has recently launched its deep learning platform called Microsoft Genomics. It has gained a lot of media attention. That is also to predict the genome regions, gene regions. And then we have a Canadian initiative called Deep Genomics. So this initiative, Deep Genomics, is mainly trying to find out new possibilities for drug development or where to target therapeutics. That's their attention. So they are very keen in identifying non-invasive drugs for existing diseases. Yeah, we are coming to that. Next comes the drug design and development. So very interesting area. It is still actively pursued by scientists. So it can be thought of as a contemporary area also, because research has been taking place for the past 10 or 20 years in drug design. But with the invention of computer-aided drug discovery, this drug design and development has had a new pace, because As you know, we are recently seeing in press like the Oxford is or has already completed the phase 1 of the new vaccine for COVID-19. So with the speed of current situation, it may take another year to come out this drug. But we couldn't think of such a kind of drug development pace in say 20 years back, because even a fastest drug could be produced in a lifespan or in a time span of about 4 years. So as per FDA's report, it took 3 to 4 years. But now, with the advent of this computer-aided drug discovery, the speed of development of drug and other related pharma products are advancing. So people are working on areas like drug repurposing. So not only drug design but also drug repurposing. Drug repurposing means utilizing an existing drug for a new disease. So for example, if we could find out a new opportunities for paracetamol, so it's a known drug. We know that paracetamol is for a particular disease. But then if we could find out new, what do you call, opportunities or new diseases for which paracetamol is effective, then that would be an amazing discovery. So they are actually concentrating on drug repurposing. Even for Nipah, for COVID also, they have tried the vaccine. They have tried some medicine for which was earlier given for that SARS, Ebola or SARS. So such kind of repurposing research is already happening. Then even these pharma giants are creating new avenues for job and career advancement. So high quality research is happening in applied bioinformatics or like this drug development and discovery. Then you have personalized or precision medicine. So this is a term that we are hearing of a new time or recent attention only. So personalized medicine refers to medical care customized to each person's genetic makeup. So it's an amazing discovery. Precision, it's also called precision medicine. So it refers to individualization of medicine. That is, what you do is just like you receive customized advertisements in while surfing the web. So Google and Amazon are using data analytics very effectively to provide customized advertisements, isn't it? While we are surfing, you receive such customized ads. So likewise, you analyze the genome of a patient, and then you understand what is it that makes him so special, and then you tailor drugs suited to that particular individual. So it means that we are saying bye-bye to the bulk assembly line drugs that we are using today, and no more side effects also. So I will be talking about a little bit more about precision medicine. So the very idea of the individualization of this medicine that people are very interested in, that's not something new, because Hippocrates, the father of ancient medicine, he was a Greek physician who lived in the 4th century. So he has rightly pointed out that it is more important to know what sort of person has a disease than to know what sort of disease a person has. So if you read this sentence aloud, you can find that this is just another way of saying that if you get to know the person better, then the disease can be understood better. That's it. So this is not an entirely new concept. So, but then the question is, how can you infer the the disease progression or the disease susceptibility by just looking at the genome? That's it. So for that, you have to first study the variations. So this personalized medicine has recently received a lot of press. So in fact, the very idea is not new, but still people have been thinking about this personalized medicine and research only less than 10 years or so. So the main revolution of personalized medicine is taking advantage of two things. That is, first is advancements in genome and the DNA science, and another one is the big data advancements, or what do you call the advancements in data analytics tools. So it's a promise. The promise of personalized medicine and big data intersection is that we get to know more about disease susceptibility, and that we can find out or we can reveal interesting patterns in the DNA, and we can find out interesting patterns from the electronic health records of a particular patient. So currently, a patient's genome is analyzed only in very critical, and only very limited portion of the genome is analyzed. even be utilized. So doctors are not currently using this genome extensively, but what we can do is we can understand the variations happening in the genome. Variations means it can have deletions, insertions, or inversions, or such kind of changes. For example, you may have heard that uh, in a particular disease called acute myeloid leukemia, the famous gene or that portion of the chromosome number 22 that is suspectably involved in this myeloid leukemia is called Philadelphia chromosome. And that funny little piece of chromosome, what it is doing is that it is overlapping into another chromosome, tail, say for example chromosome 9. So when this overlapping happens, that portion is completely overwritten. So it's simply when you think in a computer perspective, a point of view, it is simply an array of letters, A, T, C, G, A, T, C, G, G, like that. It's an array of letters or sequence of characters. So when another chromosome's characters overlap this particular chromosome, what happens? It is completely overwritten. And this have critical impact in the protein that it produces. That chromosome has been named as a Philadelphia chromosome. So maybe it was discovered in Philadelphia first. So such kind of things are happening. So that can be that can happen in 1 in a million of the population. So that kind of personalized study or individualization can help in developing targeted drugs catering to the needs of a particular individual. So, but to bring in new advances, definitely we have to have a thorough understanding of big data analytics and the tools that that aids in visualization and exploration of big data. And then comes, so I've told about electronic health records, right? So electronic health records is not a new term, but of course in foreign countries it's already practicing. So they are what do you call? They are keeping medical records systematically in a digital format called electronic health record. So, but in Kerala, even in India and Kerala, it is still not that popular. We are seeing hospitals, nurses are running here and there with a bunch of files, isn't it? From the doctor's room to the laboratory, from the laboratory carrying results to the pharmacy. So they are running with a big heap of files. Still, we are following the traditional file system. The electronic health records is not that popular, but for what do you call it? For implementing the personalized medicine or for achieving the mission of personalized medicine, the first step should be, or one aspect of that is maintenance of electronic health records. But then there comes the issue of privacy. So people are, even though they are excited by the very idea that their medicine could be customized, they are a bit apprehensive in that whether their data could be compromised. Because in the earlier case, it was the file system. So when we get discharged from the hospital, they are not going to give us the files, isn't it? They are keeping their files in the hospital itself. They are not going to give us. But now, when it comes to digital data, unless you make sure that the privacy and integrity is not compromised, it's very unlucky. So people are a bit tensed or apprehensive about the aspects or the role of EHR in personalized medicine. So the question is, can this big data be used to engage patients in their own medical care? So this is the promise of big data, personalized medicine, and big data intersection. So now with all these advancements, where are we going? So we are in the verge of seeing patients visiting doctors with a pen drive containing the whole genome sequencing information. We are telling the doctors, hi, doctor, here is my whole genome. Please take this, examine this, and suggest a personalized drug for me. Then we are not very far from this stage. But then how can we use this genetic information? That's an important question. Even though this whole genome information is quite useful, we can apply data mining tasks, but how it is going to help us to personalize drugs? So these are all some missing pieces of the puzzle, personalization puzzle. And definitely with the help of deep learning techniques and what you call the data analytics tools, bioinformatics will definitely create new tools to tailor medical care that is customized to an individual genome rather than depending on the blanket therapy that we are following now. That's definite. And bioinformatics is all set to influence life science research in a profound manner. So the buzzword of today that is has recently gained a lot of press now, personalized medicine. But still, several factors are there, as I mentioned. Several factors are there to achieve this mission. One of the aspect is electronic health record. Another one is the studying the variations in genome. Another one is the drug behavior itself, because a drug can have several the so-called therapeutic index. Therapeutic index refers to balance between the efficacy of the drug. Efficacy means how it is going to excite a person, how a person is going to be responded by that drug. So it's a balance between the drug response or efficacy and the dosage. So in what dose a particular person is excited. So the level of excitement varies from age. A kid may not, and the response of a kid may not be the same as that of an elder person for the same drug dosage. So all these are things that we studied. Then of course comes the socioeconomic factors affecting the drug response, like education, then the religious beliefs. All these, some persons may believe that particular kind of treatment is not good, or it's not. I'm not going to do that medicine, or I'm not going to use that particular treatment manner. So such kind of religious beliefs are there. So personalized medicine has to be extensively studied in order to achieve that mission of precision medicine and translational medicine. So please keep in mind that clinical applications are gaining prevalence, and unless you get updated in this area, you are going to be bioinformatics. The main excitement of bioinformatics itself that is rapidly changing at a new pace. You have always the big thing to learn, next thing to learn. So unless you get updated, it's going to be very difficult. And we are all striving hard to build novel algorithms and machines that can help the world make a better, safer, healthier place to live in. So I make an honest appeal to all of you. Let's code against the diseases. Let's code against the COVID. Let's code against cancer. Let's use our skills to develop new tools and services in the field of genomics, proteomics, and immunomics. So I acknowledge the valuable inputs that I've obtained from Professor Dr. Matthew J. Palakal. He's Associate Dean, School of Informatics, Indiana University, Purdue University, Indianapolis, USA, during his expert lectures at NIT Calicut. So I have had the privilege to interact with him closely during his visits at NIT Calicut. Some of the points I've taken are from the undocumented interactions between myself and Sir. Then I'm gratefully into the brainstorming discussions and thoughts due to my beloved guide, Dr. K. Abdul Nasir, and my mentor, Dr. G. Gopakumar, and interaction with my dear friends in the bio-research group at NIT Calicut. And I should not forget the research articles and talks by Dr. Achyut Sangares Nair. He is the Director Center for Bioinformatics, University of Kerala. So I'm an ardent fan of his talks, his technical as well as non-technical articles in dailies and media. So I'll stop by giving you a landscape of the various biological databases that contain or that amass a huge amount of biological data. So different kinds of data like gene expression data, DNA methylation data, protein-protein interaction data, pathway interaction data, variations of genome data. So all these kinds of data are available in databases, publicly available databases across the globe. So I have mentioned the websites and the links to each of these in the reference section. I'll give numbers also. So I will share the slides with you. So interested researchers can visit these websites to get information on data sets. So to mention a few, we have the Gene Expression Omnibus, GEO, by the National Center for Biotechnology Information, NCBI, Genome Browser, Expression Atlas, and then the TCGA project, which I was mentioning before. Then the Human Protein Reference Database, the HPRD. Then the KEG, Kyoto Encyclopedia of Genes and Genomes. So all these are different tools or open source repositories which help you to gather data about these different varieties. So these are different varieties of data. That's why I deliberately introduced the concept of big data and personalized intersection. So the biological data, in fact, it qualifies to be in big data, to be called big data. because it's not only in the volume, it's not only about the volume when it comes to big data. Not only the volume counts, but also the variety of data types that we have, and the speed, the rate in which information is getting pumped in. That's why we often say the 3 V's of big data. So then here are some of the top journals that a prospective researcher in bioinformatics can think of. You have the IEEE ACM Transactions on Computational Biology and Bioinformatics. Then you have the PLOS Computational Biology, Bioinformatics Journal from Oxford University Press, BNC Bioinformatics. So all these are some of the journals, top quality journals. Of course, there are several journals from Elsevier, Springer and all you can think of, but I have mentioned some top journals only. You can have several journals according to university's policy. These are the references that I took. So I would like to thank you for your very valuable time and patient listening. And of course, I'm happy to hear from you.
Thank you, sir. All the queries or questions regarding the session can be posted in the chat box. So thank you, sir, for the wonderful session.